Right, well, welcome to the presentation. Thank you for coming. My name is Nathan Huckstra. I'm a graduate student who's been working in Dr. Elias's lab the last few years. Before we get started, I want to review a couple settings. So first off, if you could please leave your video and audio off. If you have questions during the presentation, please type them in the chat, and I will tabulate and keep track of the questions and then submit them to Dr. Elias at the end of the talk. Talk today should be about 20 to 25 minutes long, and then will be followed by a question and answer session. So Dr. Mikhail Elias is a professor of biochemistry at the University of Minnesota. His lab studies microbial enzymes and systems that can be used to address industrial ecological problems, including phosphate mining proteins, enzymes capable of interfering with microbial communication, and cell density dependent expression of virulence factors, as well as control of aquatic invasive species. I'm sure he has an interesting talk for us today. I hope you find it useful. And with that, here is Dr. Elias.
Thank you very much, Nathan, for the intro. Thank you all for joining. First of all, I want to disclose that I have a dog in the background, and she's a puppy, and you might hear some weird noises, and I apologize for that in advance. She probably finds my talk really interesting already. She's a little excited. I'm happy today to talk to you about some of our project, by the way, in which Nathan is involved, and I'm going to present some of his results, where we try to suppress microbial population structure and suppress microbial communication to mitigate the spread of aquatic invasive species. And I'm going to explain you why and how this is connected. So first of all, it is important to understand that biofouling, which is, we're going to see in a little more detail what biofouling is, but it's essentially what you see, this sticky slime on any submerged surfaces that forms over time, is basically composed of bacteria, algae, mussels, larger microorganisms. Bacterial biofouling is a vector for the spread of aquatic invasive species. And in fact, there is literature evidencing that this is a very significant vector. Some papers estimate that about 70% of species, of aquatic invasive species in North America were introduced in new bodies of water because of biofouling. And this is in particular the case in Minnesota with mussels. The way biofouling is addressed currently is by using anti-fouling coatings, and those coatings are typically made of copper oxide. So copper oxide represents 85% of the market, and while copper oxide is toxic to mussels, including zebra and kaga, it is also toxic to any other organisms living in the water. And so it causes certain environmental problems and concerns. And it's in fact banned in several countries in the world, including banned in California, in discussion to be banned in the state of Washington. And it isn't overall an environmentally sustainable solution. So there is really a need for an eco-friendly solution to biofouling that in turn would be useful to mitigate the spread of aquatic invasive species. Consensus in understanding how biofouling works is simplified in this scheme here. And everything does start, in fact, with the adhesion to surfaces of biological molecules. That's really the first step. But the second step is the attachment of bacteria to surfaces. And these bacteria are going to start to proliferate. They're going to start to adhere and proliferate onto the surfaces, form a biofilm that will maturate. And this biofilm can host more specific bacteria, such as sulfate-reducing bacteria that can accelerate corrosion, for example, if the substrate here is a metal, is steel, or can favor the adhesion of larger macroorganisms such as mussels and algae. So if you wish, the bacteria here, the bacterial biofilm, they provide a glue that helps larger microorganisms to stick better onto surfaces. And some of the hypotheses that my lab came across is, can we inhibit the adhesion of larger microorganisms such as mussels by targeting bacteria, which is really what my lab was doing. And the rationale for this hypothesis is as follows. Bacteria, they can communicate with each other pretty much the same way that our body, our brain communicates with organs by synthesizing hormones that regulates their activity. Bacteria, they can produce small signaling molecules and secrete them into the environment. And they can do so in cell density dependent manner. So the more bacteria, the more of these signaling molecules, and they can sense these molecules and use this information to adapt their behaviors and adopt group behaviors. Once a certain concentration of these molecules has been reached in the environment, it will enter the bacteria, bind to a cellular receptor, regulate gene expression patterns, and so regulate group behaviors. Some of these behaviors are shown here, and they include bioluminescence, but they also include virulence and biofilm formation. So what we have is the reason why this exists is because a single isolated bacteria would have very little interest to produce and secrete molecules outside of the cell to form a biofilm. A biofilm is a very complex and dense structure of sugars and nucleic acid, and it's very costly to make in terms of energy. And so a single cell would have very little interest in spending so much energy just to have their molecule being diluted in the environment. However, when there is a lot more than one cell, then suddenly it makes sense because all these molecules are going to attach to each other and they're going to form a structure onto which they can attach. And I want to point out that bacteria on our planet, they exist mostly in the form of biofilm. So it's been estimated that 99% of bacteria on Earth are in the form of biofilm. So it's really their main form of existence. This communication here is called quorum sensing, and I'm going to talk about it quite some. So they produce these molecules, they're capable of sensing it and responding to it. And a logical hypothesis is, well, if I can intercept these molecules, if I can disrupt or degrade or remove them, then suddenly I'm blocking communication, bacterial communication. And if I block bacterial communication, I will block all of the behaviors that depends on it, okay? So this is really the rationale here. If I can block bacterial communication, I will block all of the behaviors that are depending on it, including biofilm formation. And this is what my lab has been really focusing on, characterizing what we call enzymes. So those are proteins that have a chemical activity, and their chemical activity is to degrade those molecules here, those signaling molecules. In particular, we focused on two enzymes that we improved. So we made some changes in the enzymes to improve their properties, because enzymes did not evolve to work well in environments where we plan on using them, such as paint, for example. They evolved to work well in a cell. But a cell environment is very different from paint environment. And so we had to modify them to make them work in paint, for instance. And so we verified that these new enzymes now have very, very interesting properties. They can resist aging, so they are very environmentally stable. They can also resist a variety of solvents. And so we can think of using them as ingredients into a variety of different paint and coatings. And this is some work that has been performed by Dr. Huang here he initiated this work in the Duluth Superior Harbor. He first managed to coat our enzymes and formulate it into a variety of coat. This is an example here in this top coat from Hempel. And we initially tested this coatings on steel in the Duluth Superior Harbor for corrosion. So there is a very significant microbially induced corrosion in the Duluth Superior Harbor that has been accelerating in the last 20 years. And so we thought that this was a great experimental site to test that. And so what Simon has been showing there is that if you look at this graph here on top, what you have here is essentially the degree of corrosion in the samples. And here you have different time points. So this is after 1 month in submersion in the harbor. This is after 8 months. This is after 22 months. And B is the coating alone. C is the coating plus a biocide, which is equivalent to copper. And D is the coating with our enzyme. And you see that in all of these time points, D performs the best at inhibiting corrosion, including over the time span of 22 months. Here you have the visual for it. So this is the sample B here. This is the control after 22 months. You have even fouling here. You can even see a few mussels there. And on phase B, you see more corrosion because we actually scratch that surface to accelerate corrosion. And on phase A of the enzyme here after 22 months, you see very little fouling and you see very little corrosion despite the scratching on the coating here that has been done. So we're very, very impressed by the performance and even the durability of this. Now, with the help of the MACERC, what we became interested in is to evaluate whether this technology can also inhibit more severe biofouling, including the adhesion of some invasive aquatic species. And we in particular focused in zebra mussels on sites that are known to be infested, such as Lake Minnetonka and the Mississippi River in Minneapolis. And Nathan has been performing some of these experiments where we use here coated pieces. These are polycarbonate, but we've used a variety of different substrates here. And we submerge those samples in these sites and we sample them at different times. And so some of the results here that we could obtain. So this is still the Duluth Superior Harbor, 2 different material. You can see steel. You see, so this is a control with a protein with no activity. This is the coating with copper, and this is the coating with our enzyme disrupting microbial signaling. And so you obviously see, and this is after 3 summer months, you obviously see less corrosion with our enzymes. This is polycarbonate as a substrate. Polycarbonate is a really good substrate for biofouling and for the adhesion of mussel. Here there was no mussel in the Duluth Superior Harbor. Not too surprising. It's not a very heavily fouling area, but you can still see that here you have a lot of material on the coating, and as you go towards the enzyme, you have a lot less material on the coupon. Same goes for the Mississippi River sampling. You see the polycarbonate is completely covered with algae, and as you go towards the enzyme, it's less and less covered. And the same goes for experiments in the Tonka Bay Marina, with the difference that here we had a very high number of zebras, and where we show that not only there is less algae that are sticked to our coating with the enzyme as compared to other treatments and control, but there is also no mussels that are adhering to this surface. So very, very intrigued by this performance. If you have a closer look, you can actually see in more details that here you have a clump of mussels you have a small clump, and then you just have one here. And so obviously, you know, mussel adhesion has some stochastic effect. So it's not, you know, sometimes you can have a big clump attaching to a surface, and sometimes you may have nothing. So what you need to do really is a lot of replication to try to avoid being in this trap of looking at some stochastic effect. And we're working on multiple replication. And with this replication, we actually see that there is a significant difference between copper containing coatings, and which is the standard on the market, and our enzyme-based coating, which, you know, I'm going to repeat this, but this enzyme is a biological molecule. It's non-toxic. If we look into more details, the advantage of using polycarbonate for sampling is that this material is transparent, and so we can do some imaging and actually study under the microscope to investigate what organisms are actually present. And so you can see here, in particular in the samples in Tonka Bay, you can see that, you know, you have the control with the inactive enzyme, you have the control with copper, and you have the treatment with our enzyme. You can see here that you have small mussels that are called veligers, and you have quite some here, you have quite some here, and you have none here. All right, so this is very, again, very encouraging for us. And you see a different pattern in the Mississippi River. Again, we didn't see much zebras there over the summer, but you see that there is a lot less material on the treatment. And interestingly enough, what you see is that you have a little more and bigger diatoms with this treatment. You have a lot less material on the surface, but you have a little more and little bigger diatoms. So this is something that we're currently investigating. And so the future steps in this project, what we're looking after now, with the help of MACERC, is to repeat these experiments on longer terms and with more replicates, but foremost to test it in even more real-world conditions to really assess the translational potential of this. In particular, we plan on testing this on buoys and on boats. So we plan on actually painting about 6 boats in different bodies of waters to really evaluate the potential of this as compared to some of the best coatings on the market, and decides whether this is a great competitors to current products on the market. So, you know, this is a picture of my lab. These are my lab members. I want to thank in particular Nathan and Simon Sichon here that actually did this work. My collaborator at the University of Minnesota Duluth that really helped us a lot, and that has contributed a lot to this results, Professor Hicks. I'd like to thank funders, so MinDrive, Sea Grant, MACERC, and LCCMR. And I would like to thank you very much for joining, and thank you very much for hearing me. And I'm ready to take any question of yours. Thank you. We're going to wait a little bit.
I didn't get any questions during the talk, so I didn't have any to kind of tabulate. But if people have a specific question and you just want to type, you know, I have a question in chat, then I'll unmute you and you can ask directly if you want to.
Oh yeah, we can give people some time to. I think we have something in the chat, Nathan.
Yeah, so we have a question from Kristen Toong and would like us to explain biofouling a little bit more.
Yeah, and there's really no dumb question. I mean, it's a very complex process. Maybe I can come back to my slide. Let me. Oh, okay. Okay. So biofouling is really anything that will stick to submerged surfaces. So whenever you submerge surfaces into a body of water, you will see material build up onto it. And this material buildup is called biofouling. And this material buildup starts, the first step of this buildup is the adhesion of some biological molecules that are in the water and bacteria. And the bacteria that are first building up are called early biofilm former. They are the first ones to adhere to the surface, and they will thrive on this surface, and they will start to form a sticky slime on it that will help other bacteria and other larger organisms to attach to the surface and colonize.
Right. And then we have a question, not in the chat, but in the Q&A, a question from Jessamine. She asks, is there any risk to this coating breaking down and getting into the environment?
Right, so this is a fundamental difference between the coatings that are currently on the market and this one. I probably didn't explain it well enough, so I'm really happy that you're asking this. The way the current coatings work is by leaching out copper. Okay, so copper and copper ions are toxic. And so they're going to be toxic to the bacteria that forms the biofilm. They're also going to be toxic to the mussels that adhere to the surfaces. And so, but to have this action, it needs to leach out. So those coatings are called ablative because they are designed to release constant amount of copper in the environment. This is the reason why these coatings are being banned progressively, because they are releasing the toxic molecule in the environment that obviously has a lot of consequences on aquatic species. Now, the coating that we are working on here is completely different. We are working with an enzyme that is a biological molecule that essentially is food grade. This is something that we could eat, right, and digest without any problems. And so here, it's fundamentally different because we actually don't want the coating to leach out, because every time the coating will be leaching out the enzyme in the environment, it will be eaten up by microbes and the coating will lose activity. So we want to bind and really tightly bind the enzyme to the coating so that we do not lose activity over time. So it's really the opposite of current coatings that are ablative, so they leach out over time. We want something that does not leach out over time. Difference in technology, but also difference in toxicity. Current solutions are biocidal, toxic. This solution is basically food grade.
Another question from Jane Ahrens. She asks, what else is in the coating that we use?
What else is in the coating?
Mm-hmm.
So those are very simple coatings that we worked with. Most of them are actually coatings that you can, we just bought them at your, department store. Typically, we're working with acrylic base at this time, but we are now also working with some companies so that they can provide us some of the more sophisticated polymers that they are using without their active ingredients. But really simple. Most of these coatings are simply the polymer itself. So in the case of the data that I presented, acrylic coatings. So nothing else active in the coating.
Alicia O'Hare asks, does the coating ever need to be reapplied?
Yeah, so that's a great question. This is some of the things that we're attempting to answer in our next set of planned experiment, where we're trying to assess durability better. In the longest data series that we have is this one, where we do show that over 22 months, we have a superior effect to a biocide containing coating. And so this is very encouraging. So any coating that you think of have to be reapplied at some point. What we need to characterize now is how the durability of these coating compares to the typical durability of coatings in the space that are relevant here. So, for example, if we are aiming to coat private boats, typically those coatings those boats are recoated. If you're talking coastal areas, for example, they are recoated every season or every other seasons. So typically you want the durability to be at least 2 years. This is what we're looking at. For other application, it needs to be a lot more. And this is what we're examining as well.
And I think a couple of the questions were revolving around the duration of the coating. So Mr. Peshek and a couple others, I think I'm going to count those as answered. Let me take one from Karen Chandler. Karen says, this sounds like a promising approach to prevent adhesion on hard surfaces, but we will have still many other surfaces that zebra mussels adhere to, including plants. So I'm not sure if there's what the question was there, but maybe just speaking about other surfaces.
Yeah, it is obviously true. Like I said in the intro, biofouling, the adhesion of mussels on structure is a vector for the spread of mussels to new bodies of water, but it is not the only vector. If someone decides to transfer plants, to a new body of water, unfortunately, we're going to have problems. But a lot of time is simply somebody moving a structure from one body of water to another, moving a boat that has not been cleaned properly to another new body of water, moving something to a new body of water. And in that case, it can be, ballast water contamination, but it can also be fouling and veliger in fouling. So it's not going to solve the problem, but it's going to help, hopefully.
Then we have a question from Scott Ballantyne. He asks, are bacteria required for zebra mussel adhesion in the lab, in essence, in cultured mussels?
Yeah, so this is also a great question, and it's very hard to answer this question for the very simple reason that bacteria are absolutely everywhere. And so doing an experiment without any bacteria is actually virtually impossible. And so what I do know, though, is that if you try to measure the adhesion of mussels onto surfaces, it really helps if you pre-incubate your surface in water for quite some time. In other words, it's really helpful adhesion if you let some biofouling form on your surface. So I can't answer that because essentially after 5 minutes of submersion of any surface in any water, you have biofilm forming. So it's very, very difficult to answer that, and I don't think anybody can answer. What we do know is that there is some synergy, but is it indispensable? I don't know how to answer that. I'm sorry.
And then another question from David Fisher. He asks, could this have application in power plant cooling systems or municipal water supplies that draw water from rivers or lakes?
Yes, so this is something also that we're examining. We've been in contact in a few companies. We've also been in contact with the municipality to do some testing. So yes, we are examining this. We are talking about very different durability requirements, though. Their requirements for durability is very, very stringent. It's very long. And so they are shooting for many years. And so we will need to have data collected for a lot more years before we can replace and get into this market. So it's a little more challenging market for us, but yes, absolutely.
Okay, thank you for those answers. Does anyone else have any questions that perhaps I've missed or new questions that you have? Just feel free to type them in chat and I'll relay them.
Thank you very much. Thank you, Nathan. Should
Okay.
we?
I think that is all our questions. I want to thank everyone for coming to the talk today, and thank you for your questions. If we didn't get to your question, or if you think of something else after the session, please feel free to contact us, and we'd be happy to answer questions. Yeah, and please enjoy the rest of the presentations today.
Thank you.
All right, I'm going to end the meeting now. Thank you.